Thanks for joining us for this last half of the Chapter 4 videos. We just decided to break up Chapter 4 into just two little larger videos instead of breaking it down into three small videos. I hope that's okay with you. It's a fairly short chapter. Last time we talked about the chair flip, right? This chair flips through the boat conformation, which I just realized we didn't say write down. This is the boat conformation where the footrest flips up and then the headrest flips down to do the chair flip. And when chair flip occurs with monosubstituted cyclohexane like this, we knew that we've discussed how the equatorial position is preferred over axial. Okay. Because when a big group is in axial position, there's going to be this 1,3 diaxial interaction that causes steric strain. So the question is, right, like with monosubstituted cyclohexanes, it's always easy because equatorial conformer is always preferred. But what if you have disubstituted situation, right? So let's think about that. Here's 1,2-dimethyl cyclohexane. Oops. Hope that naming is not too difficult. And can you guess whether this should be trans or cis? The two methyl groups are on the opposite side, so there will be a trans 1,2-dimethyl cyclohexane. That would be the name. Now, to translate this into a chair, remember you need to be able to draw a chair. The key idea is each bond has a parallel partner, right? And that's what it should look like. And then, yes, we will check your ability to be able to draw chair conformations on the exam, so please practice them. Okay. And the one way you could translate into the chair without making a mistake is you might want to number these carbons on the cyclohexane, and you could start numbering this as 1, this as 1, or this, any of these as 1. You just have to be consistent around the cycle in the same direction. So if we go 1, 2, 3, 4, 5, 6, we know that carbon number 1 has the methyl group down, right? That's what this means. And then the hydrogen is up. But at this position, up would be axial, down would be equatorial. At carbon number 2, we have the methyl group up. I want you to notice the up direction should not be, here's a common mistake, like this. That's not axial. That angle is not possible. Okay, that would be wrong. Here would be up, and the axial here would be down, like so. And again, this would make more sense once you make some molecular model kit. Okay, and then this is down, this is down, this is up, this is up. Okay. Remember, axial and equatorial doesn't have to do with up and down necessarily. In fact, both of these are equatorial, the way I drew them. Okay? Now, what happens once this flips? So, it would look like that. Here's carbon number 1, which was the footrest. And here's carbon number 4, which was the footrest, becomes the headrest. 1, 2, 3, 4, 5, 6. Now, at carbon number 1, hydrogen is still up, but at this position, up should look like this. Up is not this. That is not a possible bond angle. Okay, so up is this, where it's parallel to these bonds. And then down would be axial, down like so. So this is still down at carbon number 1. At carbon number 2, the methyl group should be up and the hydrogen down. And now what we notice is both of these are axial, the two methyl groups. And remember that means both of these experience the 1,3 diaxial interactions, which create steric hindrance. And so for that reason, this second chair conformation is highly disfavored. And so for that reason, the left conformer should be heavily preferred. Okay, so this is preferred. Now, interesting thing happens if you think about this cis 1,2-dimethyl cyclohexane. In this case, the two conformations will look like this. And in this case, right, like both conformations, you have one methyl group up, one methyl group axial, one methyl equatorial, one methyl equatorial, one methyl axial. And so for that reason, they would be equal in terms of energy. So there's no preference for that cis stereoisomer for 1,2-dimethyl cyclohexane. Hopefully that makes sense. Let's think about this molecule. In fact, I would encourage you to pause the video and see if you can draw the two conformations before we go over the answer here. Okay, so hopefully you had a chance to do that. This is going to be, let's see, 1, 2, 3, 4, 5, 6. Again, if you ask how do you know where to start numbering? It doesn't really matter as long as you're consistent. In fact, we could try numbering from here. 1, 2, 3, 4, 5, 6. And what we know is at carbon number 1, there's a methyl group that's up, hydrogen that's down. At carbon number 3, there's a T-butyl group down, so this would be axial, down, like that. Okay. Now, once this flips, it'll be like that, where 6, 1, 2, 3, 4, 5. Again, we just have to be consistent. What was foot headrest now is pointing down footrest. What was pointing down footrest and is now headrest after the chair flip. So at carbon number 1, we have a methyl group up. So this is now axial, which is bad, but at carbon number 3, the T-butyl group now would be axial. This would be parallel to that bond. You know you're drawing something wrong if there's no parallel partner in the structure, okay? And then the hydrogen here would be up axial. So what are we saying? This methyl group will be experiencing 1,3 diaxial interaction, but at least T-butyl is not. T-butyl is much bigger than methyl group, and so for that reason, this 1,3 diaxial interaction is very severe. Or I should say severe steric hindrance. And so for that reason, the right side conformer is much more preferred. Okay. And then I could say this 1,3 diaxial interaction is much lighter, so that's the reason. Here's a bonus question for you. See if you can solve this. Again, maybe pause the video to think about it. Oops. So if I were to do this, here's T-butyl up, next to a methyl group down, and then here methyl group up. So that's one conformation. If I flip it, methyl group up, T-butyl up, methyl group down, methyl group up. Okay, so that's the two conformations. Now this one, you just have to think about it a bit. We've got one huge group and one small medium group experiencing 1,3-dioxylsteric strain. In this conformation, it's just the methyl group. So total, I hope this makes sense to you, that the one on the right is much favored because there's only a medium grouping experiencing 1,3-dioxyl interaction. So, okay. Now that's all we want to say about that. Of course, this is the kinds of things that, like the kind of thing that you want to practice a lot with homework problems. Now, cyclohexanes or cyclic molecules can come in the bicyclic form. In this year's in this class, we're just gonna consider bicyclic structures, not like a tricyclic or tetracyclic type of structures. But here's a pretty common 3 bicyclic systems that you would see: 1, 2, 3. The last one could have 2 different versions of it. And so let's just go over those. The main thing I want you to get out of this is that you can understand what these flat structure that we draw on paper look like in real life in 3D. So let's break these down. These bicyclic compounds, these 2 molecules would be considered a bridged bicyclics. And the reason is there's a bridge here in the middle, right? And the top one has a common name called no-boronane. And this has the same structure as maybe that. Hope that makes sense to you. That's here's the cyclohexane, and here's the bridge, and of course this bridge bond is closer to us. This bond is in the back, and so that's why I drew it this way. There's a little cut or a gap here to indicate that this bond is in front, and that shows the 3D-ness of this structure. And here's the bridgehead carbons. And the way you name these is by saying bicyclo, because it's a bicyclic system. And this is a heptane, cycloheptane, or a bicycloheptane, because there's, 7 carbons. But you have to indicate how the bridge is distributed, okay? And so you could think of this as bridge, you could think of this as the bridge, you could think of this as bridge, depending on how you view them. And in this bridge, there's 2 carbons. In this bridge, there's 2 carbons. In this bridge, there's 1 carbon. So you just list those. So you say 2, 2, 1, bicyclo 2, 2, 1 heptane. Okay. Now this would be bicyclo 2, 2, 2 octane, because there's 8 carbons total, and each bridge has 2 carbons in it. This in 3D should look like this. Okay, hopefully that makes sense. Now, here's an interesting bicyclic system. You could say there's a bridge, but there's no bridged carbon in the middle. It's just bridged by a bond. There's no bridge carbon there. So we would say these are fused bicyclics, because it looks like there really isn't like a bridge carbon in the middle, but it looks like there's two cyclohexanes fused together. Transdecalin has the two hydrogens at the fused sites in the opposite direction, and this in 3D should look like so. I hope this makes sense. Here's the two axial hydrogens pointing in the opposite directions. Now cisdecalin is a little bit harder to draw. So here's the top hydrogen. I should probably draw it like so. Okay, so that's depiction of cisdecalin. Again, if you make a model of this, I think the structure makes more sense to you. Again, for this section, my main interest is that you can go from one structure to the other and back and forth. Like, you understand that this is the actual 3D structure and be able to draw them. Now, the very last topic in this chapter, we want to talk about terpenoids as a class of natural products, because they tend to have cyclic structures, sometimes even bicyclic structures. And first, you might be wondering, what is a natural product? That doesn't necessarily mean some product that somebody made that they sell that it comes from not nature. That's not a good definition of this term. In fact, the term natural product is a technical term in chemistry, and this means secondary metabolites. One way to define that is secondary metabolites are not primary metabolites. Well, what is a primary metabolites? Those are the primary things that make up our body or our structure, our cell structure. And those are proteins, carbohydrates, fats or lipids make up like the cell membrane, and also nucleic acids like DNA and RNA. Those are primary metabolites, what essential macromolecules we need to make up biological systems. Secondary metabolites are everything else that living organisms make to gain some sort of advantage. Oftentimes they're made to protect themselves, like maybe some plants make caffeine, because caffeine is poisonous to insects, so they can protect themselves by making caffeine. That's an example of a secondary metabolite. Okay. And natural products are the best source of bioactive compounds that could be exploited to develop drugs, and so for that reason they're very important. They've been the target of synthesis efforts from the beginning of organic chemistry. Anyway, so what are terpenoids? These are secondary metabolites that are biosynthesized from isoprene units. And what is an isoprene unit? Well, they look like this. This 5-carbon unit. Okay, and then what nature does is they stitch these together to make terpenoid compounds with some functional group exchange at the end. And just so we can identify these easier, we would call this the head part, and then this is the tail part of isoprene unit. If you think of this like as a fish, this looks like a tail. That's your head. And in menthol, this is the stuff that like has that cooling sensation, like it's found in cough drops and such. Hopefully you can find the isoprene units, 2 of them that make up this menthol. First, it's easy to see this head part. Now the oxidation state is different, right? This is double bond, this is not. there's a hydrogen here, whereas this isn't. Those minor things actually change as biosynthesis progresses to make menthol from isoprene units, but the skeleton remains the same. Here's the isoprene skeleton. The second one is always found in sequence right after the tail should follow the head. Okay, and so this has two isoprene units. So does camphor, by the way, and hopefully you can see the two isoprene units. Here's the head, here's the tail, and then here's, I'm just going to use a different color. Here's the head, and here's the tail. Okay, so that's a camphor, which, by the way, that's a bicyclic compound. Hopefully you can draw this in the 3D format. Right. So that's the same structure there. Okay, so for terpenoids, what I want you to be able to do is to identify the isoprene units in a natural product structure, to recognize them as terpenoids and to identify the isoprene units that make them up. Speaking of terpenoids, steroids are an example of terpenoids, and you might have heard of them because we actually make these natural products. They're usually hormones. Okay, and you can tell that something is a steroid because they all share this common tetracyclic structure. And yes, I want you to be able to recognize the steroid skeleton like this. Here's an example of steroid. R group, that means the rest of the structure. Methyl, hydrogen, 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 methyl, double bond, and a ketone. And if the R group is alcohol group, that's a testosterone. Testosterone. Okay. That's the male hormone that like makes us more muscular or whatever. And then if the R group is this ketone compound, or acetyl group technically, then that is a progesterone, which is a female hormone that makes you more feminine in biological features. Now, I want you to think about this, just how interesting biomolecules are. The difference between an alcohol and a ketone functional group and the rest of the structure is the same. That's the main difference between male and female, chemically speaking. Okay, very close, very similar, but the biological system is, of course, able to recognize the difference, and that makes all the difference. So that's why drug development is a really difficult task, because even a tiny, and also a promising field, because even a tiny difference in the structure of the molecule will be seen very differently by the biological system. And I want you to kind of be able to see that, like, this might have the isoprene units. Now, methyl groups and whatnot have moved around, and so you might not be able to see the isoprene units completely well, and that's because steroids usually come from this linear molecule called squalene. Okay. Here's oxidosqualene, because one of the double bond has been oxidized to this epoxide or this functional group. And hopefully it's pretty straightforward for you to see all the isoprene units in this linear form. And what happens here is mechanism wise, this thing pops up when following an acid, which is followed by these cascade of rearrangements. And that's what gets you this tetracyclic system. Okay. And after some more steps of rearrangements and functionalization, this becomes the steroidal compounds, and that's the biosynthesis of this. Okay, and that concludes the video lecture for this chapter. Sorry for the longish video, but I'll see you in class.